Hello, everyone. Welcome to episode 3 of ALS Clinical Trials Unboxed, brought to you by ALS Canada. My name is David Taylor. I'm the Vice President of Research and Strategic Partnerships, and I am super happy to be able to introduce you to our guest speaker today, Professor Dr. Richard Robitaille, who's a professor at the Université de Montréal. He's a longtime member as a board of directors at the SLA Quebec, a scientific and medical advisory council member at ALS Canada, and just like if I were to introduce this individual, this is somebody who was a world expert on neuromuscular junctions, who found his way into ALS and ended up getting funded for some things that really built into an incredible story. And he has engaged around the world, built collaborations, and really just done an amazing job. So it's super exciting to be able to have him here today to talk about where that research has led to, which is a great clinical trial that we're very excited we'll be recruiting soon in Canada. So without further ado, I'd like to pass off to Dr. Richard Robitaille. The floor is yours, and thanks for joining us.
Well, thanks so much, Dave, for the great introduction. I think this is too much. Hello, everyone. Very nice to be here. So I will start sharing my screen. Just a second. I'll get myself organized. There we go. This one and this one. So do you see the full screen okay? Is that good?
Yes, we do.
Okay, excellent. So thank you so much, Dave. So yeah, in the next 10, 15 minutes, I'll present to you a sort of I guess an achievement for a research lab like ours to be able to bring our effort to a clinical trial. So its short name is ALS Diary 1, but the full name is a phase 2 study to assess the safety, tolerability, pharmacology of 15 milligram darifenacin daily in patients with ALS. Somewhat different than probably you're used to have is this is not a drug company trial. It's really a trial that's originated from an investigator. So it's called an investigator-initiated trial. And you'll see in the story that I'll bring up to explaining the trial, I think this was important. So how does it work? Well, you're probably very familiar in ALS with motor neurons, so the neurons that control the movement. So let me put a pointer. There we go. All right. So you're familiar with the brain making decision, deciding to make a movement. That command will go to your spinal cord, and there's a relay neuron, another motor neuron, that will actually have these long connecting fibers, these axons, reaching to the muscle and make the movement. Well, that is not done on its own. It's actually mediated through a structure that's super important that they've introduced called the neuromuscular junction. So neuro for nerve, muscular for muscle. So junction is it's basically linking the nerve part with the muscle part. So it's really a nerve muscle contact, and I'll use that for my presentation today. And essentially, they're fundamental for the control of movement. So whatever the brain decides to do, it has to go through these contacts. And they're very specialized, and your muscles are made of thousands of these fibers that contract to make the movement, and each individual fiber are connected by one unique, very special of those nerve contacts, neuromuscular junction. Now, why am I saying this? Well, in ALS, of course, you have the neurons that are sick. Eventually, they will degenerate, they will die, and of course, if they die, you lose contact and you lose the control of your muscles. But we know also that in ALS, these nerve muscle contacts actually are altered and degraded way before neurons start dying and before even symptoms appear. And we know that from patients, but also from animal models. And what's very, very important is that this alteration in the neuromuscular junctions, in the nerve muscle contacts, is ubiquitous in all forms of ALS. It's always happening in patients and in mice. So it's fundamental for the disease process. So a bit more than that. So these little junctions have a compartment on top of them that regulates their function. There's some sort of support cells. We call them glial cells, but they're support cells, and they're super important for their function, which is to maintain them and also to repair them. Because in the nervous system, if you bump your muscle, for example, in the corner of a table, you will have an injury, but it will fix itself, and the system can do that. And these support cells will do that. That's what they do. They will make the system, this nerve contact, very efficient, but it will repair it if it's damaged, if it's injured. And that's regulated by a master valve. That's why I have this here as a cartoon. These little cells are actually regulated by a type of receptors. We call them muscarinic receptors. They're receptors for a neurotransmitter substance, acetylcholine. It's a signaling system in the nervous system. And what these do is basically they are really the master chef that will decide whether the cells can go into the repair mode or in the maintenance mode. Okay, so that's the normal thing that we've shown in the lab. In ALS, things go crazy. So this balance is no longer done properly, it's no longer executed, and it leads to really maladapted nerve muscle contact. So we've shown over the years in our lab that these little support cells are super important for this function, and they are actually maladapted, not normal in ALS, and it causes this instability and destruction of the neuromuscular junction in the eyes. And how's that? Well, we know these muscarinic receptors, this master valve, are hyperactive. So they are really, really pumping up the system way too much. And so the image that I have there on the right, I think it really explains really well what's happening at the NMJ. So these are our cells that are normally there to listen to what's happening and decide whether they need to repair or maintain, but they don't really understand what's going on because they have their headphones on their ears, heavy metals, hard rock music, decibels destroying your ears, and they don't understand what someone beside you would actually be screaming of. So they're oblivious of what's going on at the nerve contact. And so they cannot do this balance. So our hypothesis was if we restore that, if we bring these muscarinic receptors to a level that it's more normal, can we actually restore that balance and then restore the functional aspect? And so by using an antagonist, a blocker of these receptors, in principle, we should improve the function of the nerve muscle contact and then have a benefit on autonomy, quality of life of ALS patients, restoring their locomotion, muscle movements. So if I take the same image, suddenly the guy can listen to the secret that the nerve contact would actually tell them and realize, oh my God, I need to do something, and then restore the balance. So we basically want to remove the headphones from these cells, restore the activity, and then let these cells decide what they need to do. So what we decided to go on with this is to choose this drug, darifenacin. It's a blocker, one type of these muscarinic receptors. It's M3, so it's one of those types. It's already approved by Health Canada, FDA. It's approved in Europe also for the treatment of overactive bladder. And so essentially what we're doing here is drug repurposing. We're taking a drug that's used for another purpose and we're using it in ALS. There are a couple of reasons why we choose darifenacin. Super specific, very little side effects, and it doesn't cross the barrier that protects the brain that we call the blood-brain barrier. So the drug doesn't go into the brain, limiting the side effects. So, and we can come back to that if you want. So the idea was, if we use this blocker, darifenacin, We reduce the activity of our master valve, or muscarinic receptor. We help the cell function to be restored, and we repair the nerve muscle, and in principle, help movement, muscle strength, and so on. So that was the idea behind it. That's how we got there. I'll show you briefly the results that we had in the animal models, and then I'll move to the clinical trial after that. So if you basically, we did is to use animal models like mice in this case, and you treat them essentially as if you would do in a clinical trial. So when the symptoms would come, we would give the drug to the mice, and then we would treat them for a long time and see whether there would be any benefit. So here what you see is a cartoon of a muscle fiber, and in red you have the site on the muscle that received the message, right? The receptors that receive the message. In green you have the contact from the axon from the motor neuron. So this is our neuromuscular junction, our nerve muscle contacts. This is a well healthy contact that you see green on red, and this is one that has no green, so it's denervated, so it's broken apart. And long story short, the drug darifenacin improves the proper contact, the innervation. So it makes the contact better, which is exactly what we wanted to do. It improves muscle function, but a little video will show you much better. And here you have a mouse that received the vehicle, and here is a mouse treated with our drug darifenacin. So that little mouse here will be put on the bottom of the cage, and you see right away this poor thing is paralyzed. You see the hind limb is actually not moving. The mouse is tiny; it lost weight a lot, and so that's a very sick mouse. And the mouse is carrying a gene mutation that makes ALS come up into the mouse. And this is a mouse treated with our drug darifenacin. So you'll see the mouse wobbling around, walking around in the cage, sniffing around. It's going to stand up on its hind limbs, just like a regular mouse would do. And it's also fatter than this one. So it definitely improves locomotion, it improves movement, it improves muscle strength. And we also saw at the end that the survival of these mice were extended by about 2 weeks, which is a good duration for that type of study. So equipped with that, we basically teamed up with clinicians in Montreal, but also in the States and colleagues in Europe. So I'm the team leader on that. Jonathan Glass, a super ALS trialist, neuromuscular expert at Emory in the United States in Atlanta. Dr. Gange at McGill University, and Dr. Sandrine Dacruz is a molecular biology specialist in Brussels, in Belgium. And so the trial came up from the lab. So we had to put this team together and look for funding. And we based on these data and the quality of the clinical team, we were fortunate to obtain funding from ALS Association from the US to support that. Some funding from the MNI as well. And there is another funding that will have a second site. I'll come back to that in a minute. But I cannot disclose it right now. It's still under embargo, but we have a second site in Ottawa. So Dr. Gange and Oliver Blanchard will be the sponsors representative. They will be managing the trial. Dr. Blanchard being the principal investigator. These are the team management, Nisha and Gabriele, they're fantastic. And as I mentioned, we were also along the way, we managed to secure additional funding to have a second site in Ottawa, and I'll come back to that for the importance of that second site. And also, we were also very fortunate to have been able to convince Apotex, who's the manufacturer of the drug, to basically do an in-kind. So basically, they give the drug for the study for free. And so we have the drug from Apotex to do the study. So what is the trial? How is it? How would we decided to do? This is the little pill. It's a little white pill, darifenacin 7.5 milligram. So it's a randomized, double-blind phase 2 study, 2 arms, which is essentially with the main objective to study the safety, tolerability, pharmacokinetics, and dynamics of darifenacin in patients with familial and sporadic form of ALS. So the participants will take daily by mouth 7.5 milligram pills for 2 weeks, and then increase to 15 milligrams. So they'll take 2 pills after 2 weeks of either placebo or darifenacin. So we have 2 sites, as I mentioned, so Montreal Neurological Institute, and Ottawa with Dr. Ari Briner. 30 participants per site, and it's randomized 1-2. So we will have 10 patients on placebo and 20 patients on drugs per site with the age here. And I'll give you a bit of the inclusion criteria, but I have an extra slide if you want to see the details of the inclusion criteria. Anticipated start, it's very soon, Q3 of 24. In fact, we're hoping to get the final shipment of the drugs in about a week or 2. So in principle, we're ready to go in Montreal. So hopefully maybe in July for the MNI site, and for the Ottawa site, which we just got initiated, is going to be offset by a few months. We're hoping to be ready around October in Ottawa. So how does it work? What are we looking for in this trial? So as I mentioned, we were looking for familial and sporadic form of patients, a maximum of 36 months from symptoms onset, the forced vital capacity, so the respiratory functions of 50% or more. And of course, all patients will be able to continue their standard care treatments. So riluzole and darivone, that's actually available in Canada. And I can come back to that when we talk about possible combination of treatment. So our primary outcomes, I mentioned that several times already: safety, tolerability, pharmacokinetics. We're quite optimistic about that. The drug has very little side effects. There's no obvious interactions with drugs that are taken by patients, riluzole and darivone. So we're very optimistic about our primary outcome. So that will be that patients will be able to safely and take the drug, and it will be tolerated, although we need to do that. The secondary outcomes are we're very excited about. So we're approaching this with a new vision, which is targeting the neuromuscular junction. So our secondary outcomes and our exploratory outcomes will be very influenced by looking at the measurements that will reflect the function of the muscle. So in particular, we will use here the handheld dynamometry, so essentially measuring the force that someone can generate. And also this one here, this is called, it's sort of complicated name, electrical impedance myography. Essentially, this one will tell you the quality of the muscle, how healthy is the muscle, in addition of the force it can generate. Now, the second site is super important because it allows us to have a sufficient number of patients that these 2 parameters will have enough power that we will be able to say whether our treatment has any potential effect or not. So we're very excited about that because we can have in a few years a possibility to be able to say whether this is a good idea or not a good idea. And finally, in the exploratory outcome, I won't go through all the list here, just to point out to you again that we're looking at neuromuscular markers. So how the neuromuscular junctions would work, how the muscle would work. And we're looking also at their neuromuscular junctions themselves, because we can do biopsies and we can look at the neuromuscular junctions. So these 2, the neuromuscular junction biopsies and the single fiber EMG, they're more sort of invasive approach, and these will be on voluntary basis for the trial. But it's very exciting because we can direct measurement of the target of our clinical trial, which in ALS is actually very rare. So we're very excited about that. So this is the schedule of the trial. So you first come talk to the neurologist. You're included. Then you come a few weeks later, and you see it's a sort of a busy schedule. It's sort of a demanding trial for that. The first visit where you're randomized, whether on placebo or drug. The first few days where there's a number of tests that are done. The optional visits, that's where you would have the more invasive techniques if you choose to have those. And then after 2 weeks, you go from 7.5 to 15 milligram, and then we continue the visits all the way until the end. So that's the trial. So in summary, um, we're very excited for that because we are actually on a novel therapeutic target in ALS, which is the neuromuscular junction, so the nerve-muscle contact. So it's totally different from what's being tested now in ALS. So let's see if whether this is a good idea or not. Again, as a uh, reminder, the primary outcome is safety tolerability. So whether it's safe for patients to take this drug. As I said, we don't expect much problem there, but it needs to be done properly. We're very excited about the secondary outcome, owing to the second site that increased the power of the study. And at the end, we will be able to tell the community whether our idea is worthy of continuing and testing further or not. So we'll have a good indication on that. And as you could see from the mouse data, what we're hoping to achieve is to maintain a better muscle strength, so a better muscle integrity, which would bring better motor functions for patients and hopefully improve quality of life for patients. So independence, pride, and so on and so forth. So here, really, we're just about ready to go in terms of recruitment. So with people living with ALS, within 3 years of diagnosis with muscle weakness and wasting upper lower extremities and can swallow tablets. That's one limitation. It's a pill, so you have to be able to swallow it. This is the information to contact the clinical unit here or the phone, and you can use this QR code if you can reach out. And then just acknowledgements. Of course, we're very grateful to Apotex for giving the darifenacin, the darifenacin tablets. For that, it's super generous and helpful. Of course, the funding sources, without those, the trial would not happen. Again, it's an investigator-initiated trial, so we have to come from these sources of funding. And there's another source that hopefully soon I'll be able to publicly indicate. So I will go back to the previous slide in case for keeping the summary there, and I'm happy to take any questions.
Thank you very much. That was great. And we have a action-packed 6 minutes of Q&A that we're gonna go through here. I wanted to we do have a few questions that came in, and actually one of them that came in from a couple of people in advance was about Ottawa. So I think you answered that, which is fantastic because I know there's been people wanting more opportunities to participate in research in Ottawa. So it's great that this will be in Montreal and Ottawa as a second site. So one of the things I wanted to quickly just get your thoughts on. So when you present this, it comes off very much like you had this idea and it went to clinical trial. But this was a 10-year odyssey that came from, you had this idea around the perisynaptic Schwann cells, the glial cells, and then you explore it, you find the next step, you talk to the right people that allow you to come from a very early idea as a fundamental researcher to the point that you're now talking about on a webinar like this, a clinical trial. Like that's an amazing translational 10-year thing.
Yeah.
Did you want to maybe just expound on that a little bit?
Absolutely. So the whole thing started somewhat as a sort of a serendipitous observation from looking at the neuromuscular junctions in these ALS mouse models. But based on our background at the NMJ, we right away saw the significance of that observation. That showed us that these little glial cells, these these support cells were actually wrong. So, and then you make your way through. You work, you test your own hypothesis in the lab. You see, yes, okay, this is how I think it works. So that the muscarinic hypothesis came up. So we tested that, but very early on, and that's, I mean, the ALS Canada community, but in general, the ALS community has been extremely supportive and generous of that, is you have these opportunities to talk to clinicians, talk to other colleagues, other researchers, and then you create this network. And very early on, and when we started to think seriously about could there be a translational aspect? Could we improve? Can targeting the NMJs will actually improve? We could actually talk to clinicians very early on, and we were guided by them in terms of how to design this experiment, what they were expecting, what are the pitfalls, what to be careful about, because as you said, we're basic scientists. I've never come close to anything like that in a clinical trial. So this was extremely important. And that process took indeed about 10, 12 years. So from the first grant funding that we received from ALS Canada, actually, um, up to the trial, obtaining the funding for the trial, it was 10 years, 10 to 20, 12 years. So it was a gradual, multifaceted process of doing our homework in the lab, talking to clinicians, and designing this idea to test it.
Great. Yeah, no, that's fantastic. And so we actually have a number of scientific questions that came in. What we're going to do is, if you're okay with it, afterwards, I can send you some of the questions that came in,
Yes,
and we can get those answers out
absolutely,
to the individuals that had them. So
absolutely.
one of the things I should say, I absolutely, people will tell me I didn't ask, and it's one of the things about investigator-initiated trials and how you have to try to find funding for these things through grant mechanisms and stuff, is that, an open label extension is something that becomes really difficult because it's extra cost. People have to come into clinic to get monitored and stuff. This would be something that would 100% be considered if you were to move this to a pivotal trial. Is that right?
Yes,
Yeah.
yes,
Yeah.
absolutely.
And and
Yeah.
and and
So
yeah.
yeah, and in this, it's sort of we it's somewhat particular, is that you design the trial not on. I mean, I think these are good. This is a good design for a trial. This is not my expertise, but I think I trust very much my clinical colleagues on that. It's a good trial, but we're limited by the funding. So we have to design the trial based on the amount of money that we can get,
Yeah.
and not the reverse. Not, okay, I've got all this money, I will design the best trial. No, you have to do the best trial based on the resources that you have, which is a different way of approaching a trial. That's the limit of the investigator-initiated trial.
Well, that's a good point, and we should talk about it anyway, just to see what opportunities there are out there, even as
Absolutely.
ALS Canada. So I have one last question that we have time for, really quickly. Neurofilament is a big thing right now that people talk about, but the unique thing about this one is it could be something that, if it were early and actually triggering to neuronal death or neuronal pathogenic mechanisms, you might say you're going to rescue neurons by doing this. But at the same time, if neurofilament didn't go down, that doesn't mean that it's not working in a sort of like a while the neuron is still sick, it's providing it a better structure, to actually a better ability to do its job. So like that one's really interesting, right? Because it may
Yeah,
not tell you either way depending on what the neuromuscular junctions role is. Is that
yeah,
right? Or
yeah. No, absolutely. So there are different ways of approach analyzing or understanding the change in neurofilaments. I'm on you with this. I think it's one aspect, and what we're and I went through this very quickly, is the trial is also designed so we can actually look at different proteins, different components of the neuromuscular junction as biomarkers. So we can actually, because it's early, it's happening outside of the central nervous system. So whatever is changing in the muscles will end up in the blood. So we can actually monitor that. in combination with neurofilaments. And then we can, I think we can make a lot more sense of these changes based on these fundamental change and this very fundamental contact between nerve and muscle.
Yeah, and huge props to you on target engagement. That's the biggest thing we need, and you're gonna have it in spades through this. Like there's a lot of targeted, the EIM, all of that's amazing. So we're actually already out of time. I will tell anyone that the questions we can get, we'll try to get some answers for you. If they're questions that weren't directly related to this, please do come to our ALS Research and Clinical Trials 101. We can answer those or discuss those different things. And we will do our best to get the answers back to everybody who did have questions. But thank you very much, Dr. Robitaille, for your time today. This was incredible.
Thank you for having me.
And please, look for the trial as it comes out. And if you're in the area and you can participate, that's wonderful. And thanks again to everybody.
And any questions, please send them over. I'll be happy to handle them. And if I can't, I will find someone that will be able to.
Perfect.
Great.
Well, thanks again, and thank you to everyone. And we'll see you again at in a couple of months at the next one. So thanks, and everyone have a great day.
Have a great summer.